Hello, this is Gina Piscatelli with the third lecture on muscle tissue and muscle physiology. In this lecture, I'll talk about the structure of individual skeletal muscle cells. So before we get started, let's just review what we already know about the structure of whole skeletal muscles. We know that an entire skeletal muscle, an individual one I should say, is covered by a connective tissue called epimysium, and that the muscle can be subdivided into individual groupings, each one called a muscle fascicle that's covered by connective tissue called the perimysium. Now, even a muscle fascicle can be broken down into smaller subunits, and those subunits are called myocytes. So that's the cellular level. Many myocytes make up one muscle fascicle. And each myocyte is surrounded by a thin connective tissue called endomysium. The structures that I'm going to talk about in this lecture that are inside of a myocyte, well, I guess part of a myocyte, will be the cell membrane, which we call the sarcolemma. We'll look at how many nuclei are present in a skeletal muscle cell. There'll be many of them. And of course, I'll point out again the myofibrils, composed of individual proteins called myofilaments. Inside of a muscle cell, the endoplasmic reticulum functions a lot like any cell we've talked about. We have a special name for it though. We call it the sarcoplasmic reticulum, and we're going to see that it's important for calcium storage. In addition, we'll note that there are multiple mitochondria present in muscle cells, and that there's a structure called a triad, which is made up of terminal cisterna and transverse tubules. We'll talk about what those are in a little bit. And then finally, we'll look at histology a little bit and recall the striated pattern that you see in skeletal muscle tissue. And the repeating subunit is called a sarcomere. And the sarcomere is really also the functional subunit of a muscle cell, because when contraction occurs, it's the sarcomeres that shorten. I'm sure a lot of that doesn't make sense to you yet, so we'll go through it stepwise. First, I'll start with the cell membrane is called the sarcolemma. And you can see I've got one myocyte on this picture, surrounded by the cell membrane, which is just a phospholipid bilayer, and we call it the sarcolemma. Inside the cell, there are many nuclei. This particular picture shows a cell with 3 nuclei. And the reason there's more than one nucleus in a cell is because during embryonic development, cells fuse together to form a bigger cell. Now, within the cytoplasm of a myocyte, there are myofibrils, which are groups of contractile proteins. So I have one myofibril pulled out of this cell, and you can already see a striated sort of pattern here. Each one of those subunits is called a sarcomere, which we will come back to later. Now, frequently I'm going to use this picture instead of the one that's in your textbook. The previous slide contained a picture from your textbook. This is from a different textbook, but I like it because it's I don't know if more detailed is the right, it's just three-dimensionally, it seems to make more sense. So on this particular picture, we still have the same structures as before. The cell membrane is yellow, or the sarcolemma. There's more than one nucleus. There's a purple one here and a purple one here. And again, there are multiple myofibrils in the cytoplasm of one myocyte. And I've got one circled in black in the lower left. Now let's add some more structures. And that will be the myofilaments and the sarcoplasmic reticulum. So first, the myofilaments that are located inside a myofibril. There's two types. There's thin ones and thick ones. The thick ones look purple and the thin ones look, I don't know, orange or salmon colored. And these are the contractile proteins that are bundled together into a grouping called a myofibril. In between the myofibrils, there are many mitochondria, which I'll show you another picture shortly. And the endoplasmic reticulum weaves in between all these myofibrils. We call it the sarcoplasmic reticulum, and it is blue in this picture. Okay, and then finally, I want to note that there are multiple mitochondria, and the importance for this is that you remember that ATP production occurs by the Krebs cycle and the electron transport chain in the mitochondria. So skeletal muscle cells need a lot of ATP for contraction and relaxation, and so there's multiple mitochondria in the cytoplasm, kind of wedged in between all these myofibrils. And then the last couple of structures form what I call a triad, and your book calls it a triad as well. The transverse tubules are deep extensions of the cell membrane. So although the cell membrane in this picture is shown as yellow, they've made the transverse tubules purple for some reason. I don't know why. But the transverse tubule, you can see here, kind of comes up and attaches to, or is really part of, the sarcolemma. And it's repeated throughout the cell. And then the terminal cisterna, which are colored blue in this picture, are part of the sarcoplasmic reticulum. So those are like tubes periodically occurring along the length of the cell. So here's one triad, here's another triad, and that continues sort of a doublet pattern like this down the length of the myocyte. Here I have a triad kind of defined and labeled. Triads consist of 3 tubes that are grouped together. That's one transverse tubule, which is purple in this picture and is part of the cell membrane, or extensions of the cell membrane, the phospholipid bilayer. And then also 2 terminal cisterna, which are kind of end points of a sarcoplasmic reticulum, kind of. Now you'll find these triads on either side of a structure called a Z-disc. And the Z-disc repeats down the length, through the width, and then recurs down the length periodically throughout this myocyte as well. So in your textbook, this is what a triad looks like. 2 terminal cisterna that are blue with the white transverse tubule in the middle. And then the Z-disc is kind of off to the side of that. And there would be another triad on this side of the Z-disc over here. The structure from Z-disc to Z-disc is called a sarcomere, and we're going to talk about that in the second part of this lecture. First, let's just review using this diagram that I found that I think is really cool. So this shows one cell, a myocyte, with individual myofibrils, and then if you enlarge it, this is what I think is so cool about this picture. If you enlarge it, you see that there are mitochondria located in between all the myofibrils. And there are also extensions of the cell membrane and those terminal cisterna, which are extensions of the sarcoplasmic reticulum, all located in between the myofibrils. Three-dimensionally, I just really liked this picture. I think it's really cool. Okay, now we're going to go to the second part and talk a little bit more about these myofilaments. So myofilaments are either thick or thin. The thick ones are called myosin, and they're purple in this picture. And the thin ones are called actin, and they look kind of orange or salmon colored in this picture. These are contractile proteins. So they're going to actually slide across one another longitudinally or lengthwise to cause this myocyte to shorten during muscle contraction. To understand how that works, we need to look a little bit more closely at the structure of myosin and actin. So myosin filaments are made up of several myosin proteins, which are dimers, meaning there's two myosin proteins kind of coiled together. The tails are coiled together, and then the myosin protein has these two heads. We call them globular heads. Now these heads can bind actin, which they will do so during muscle contraction. And they can also bind ATP or the breakdown products of ATP. So myosin is the one that has the heads, the globular heads. Actin looks more like a straight filament. Okay, actin is again a dimer. It's made up of two strings of actin subunits. They're kind of coiled together like two pearl necklaces. Now, actin subunits have a binding site for myosin. So that's where the globular heads will bind. Covering those binding sites are two proteins called tropomyosin, which is orange, and then troponin, which is yellow and kind of keeps that tropomyosin in place. Okay, let's look at, kind of go backwards and think about histology in terms of skeletal muscle tissue. You can see the repetitive pattern, the striations. Okay, those striations really reflect the organization of those two filaments, myosin and actin. We call the repeating subunits sarcomeres. So I've kind of blown the picture up over here on the right and showing that there's a light region, then a dark region, then a light region, then a dark region, right? In the light region, there's a thin dark line. That's our Z-disc. And so a sarcomere extends from one Z-disc to the next. And so what I've blown up or magnified shows 6 sarcomeres here. These are the contractile subunits of a myocyte. And so they will actually all shorten, kind of scrunch up together during muscle contraction. Now the reason that the sarcomere has these light regions and dark regions is because of the arrangement of the myofilaments. The dark regions are going to have both actin and myosin overlapping, and the light regions will only have actin. So from Z-disc to Z-disc, this picture's from your book, in this dark region in the middle you're going to have both actin and myosin, but the light region will just have actin. So here's what the arrangement looks like schematically. We have one end of a sarcomere is where the Z disc is located, and then there's the other end of the sarcomere, a Z disc. That Z disc is made up of proteins. One of them is alpha-actinin. You don't have to remember that. And the actin filaments in this picture are red, and they are directly attached to the Z disc. The myosin myofilaments are purple in this picture. They're thicker, and they sort of appear to be suspended in the middle, right? And then there's this protein in the center that kind of keeps them all together, because you have to remember this is kind of actually a three-dimensional structure, the sarcomere. It's cylindrical, and it's important to keep the myosins in place. So when we look histologically and we see dark regions, we are seeing myosin and actin overlapping. The actin overlaps with the myosin. I can go back to this picture, one we were looking at before. So this will be the dark region. There will be a little tiny light region in here, but it'll be obscured. You won't be able to see that. And then there's a light region over here. So light and dark. Okay. Now we actually label these dark and light regions with terms. The light regions are called I bands. I don't know who named them or why. And the dark regions are called A bands. So I remember I band for light, Li, and that has only actin, which sort of makes sense. And then the A bands are dark, and I remember that because dark has an A in it, or dark. And it's going to contain both actin and myosin. Now, within that A band, there is the region that I mentioned that contains only myosin, because the actin doesn't extend all the way across. It comes from each Z disc towards the middle. This is called the H zone. One thing to think about is when these myofilaments interact, they bind together. I'll go back, sorry. When they bind together, the Z disc is going to move in that direction towards the center. Both Z discs will, because the myosin is going to pull the actin filaments towards the middle. So what's important is that all the myofilaments, all the muscle cells, all the myofibrils, the whole muscle, do this all at the same time. So everything has to be connected somehow to the cell membrane, or else it would all be disrupted or fragmented. So there are some anchoring proteins. One in particular is called dystrophin. And dystrophin is shown as red here. I just put a blue line on it. And it anchors the superficial actin filament to the cell membrane, so that the cell membrane moves with all the contractile proteins. So everything's going to shorten together and not be pulled apart. Dystrophin is a protein coded for by a gene. And when that gene is mutated, we have a disorder known as muscular dystrophy. And so people are unable to contract their muscles. There's also an anchoring protein called titin that keeps the myosin in place. It's a titin and elastin both keep the myosin evenly spaced between two Z discs. Now, if we think about a sarcomere three-dimensionally, we really have to look at a whole myocyte. So this shows a cross-section through a myocyte at the dark region where the A band is. And you can see that one myosin, which is the purple, is surrounded by several actins. So one myosin can pull on many actin filaments at the same time. If you look at an I band, you only have actin, and if you look at the H zone, you only have myosin. The last thing that I kind of want to mention to you before we do a little review is that, skeletal muscles, not all of them, but many of them are elongated. And, at the ends, we've got two tendons, right? And so when this muscle contracts, it shortens, right? And actually, it's going to shorten from both sides, and the belly gets thicker. Now we have to think about what we just learned about sarcomeres and what the orientation of the sarcomere is and the muscle cells. So the muscle cells run lengthwise. So the muscle cells run longitudinally within that muscle. Those muscle cells need to shorten. And so therefore, the striation patterns are perpendicular to the muscle length. So I'm going to outline in this histology one myocyte, and that's in the proper orientation relative to the orientation of this muscle. So the myocyte's running lengthwise, but the striations are perpendicular. Okay, they run kind of the width, so to speak. And that's because, as you know, the sarcomeres are going to shorten kind of towards each other. It's going to scrunch. The whole myocyte's going to get shorter. Okay, now the last thing is not new information here, but it presents, I'm going to present an idea for you in studying this, and that is that to understand muscle contraction, it's really important to know the structure of a sarcomere. So I would practice, if I were you, drawing a sarcomere, starting with two Z discs that I've drawn in this picture. And then attach your actin filaments, which are thin, and they don't come to the middle, right? There's a blank spot in the middle. And then fill in your myosin filaments, which are thicker. And now you can do some labeling, and you can be thinking about function a little bit. So the first thing I would do is draw some myosin heads in. Okay, so you got to put some globular proteins or heads on the myosin. Okay, that's my attempt at drawing myosin heads because they're going to grab the actin. And then you want to be able to label the I bands, the A bands, and an H zone. And make sure you know which is lightest and darkest, right? And identify which one only has myosin, of course. So here's the answer for all of that. But I want you to practice on your own, of course. So the I band will actually encompass two sarcomeres. The Z line is in the middle of an I band. And then an A band, which is the dark region within the middle of one sarcomere, and then the H zone or H band where you only have myosin. And then also present on these slides, I'm just going to let you know that I posted this exercise. It's a picture of a three-dimensional sarcomere model, and you can print it out if you want to and practice labeling, or you can just look at it and do it or whatever. We obviously don't have these to give to you, but they're kind of made out of plastic and wood, and it shows two Z discs at the end of a sarcomere. It shows the middle H zone, and then it shows the dark region, which has both actin and myosin, which we call the A band. And then over here we have a light band with only actin, and then this is the Z, right? And the actin and myosin are also labeled, which I can let you do. You can see these little boxes here. Okay, so our next lecture will be about the function of this sarcomere during contraction. That's all for this one. Thank you.